Hello and welcome to the biochemistry presentation. This will be presented by myself, Kyle Winship, and Brian Thompson. Today we'll be presenting on the research article entitled Signaling Complexes Control the Chemotaxis Kinase by Altering its Apparent Rate Constant of Autophosphorylation. The significant autophosphorylating histidine kinase involved in chemotaxis of bacteria, Ka, has been shown to change its kinetic behavior of autophosphorylation if it is embedded in a signaling complex. Ka donates its phosphoryl group to the enzymes Ky and Kb, which are responsible for flagellar rotation and sensory adaptation, respectively. As seen by studying Ky phosphorylation, kinase activity is affected by its incorporation into signaling complexes by a factor of up to 1,000-fold. In addition, it can be affected by ligand occupancy and an adaptation modification. The activity of Ka controls the concentration of the active form of the response regulators, Ky and Kb. Integration with the signaling complex controls this kinase's activity by changing its rate constant. When Ka is isolated, it has a low kinase activity. However, when it is incorporated with signaling complexes with chemoreceptors and Kw coupled with the phosphorylation of Ky, it has a dramatically increased kinase activation up to a factor of 1,000-fold. The study by these researchers investigated how enzymatic kinetics were affected by integration with a signaling complex by applying steady-state kinetics and Michaelis-Menten relationships to the autophosphorylation reaction of Ka. Researchers also studied these kinetics in three contexts. 1. Isolated Ka. 2. Ka incorporated in isolated intact signaling complexes. And lastly, Ka incorporated into signaling complexes of small arrangements. Figure 1, shown here, depicts Ka and shows how it has 5 domains labeled P1 through P5. P1 is where the substrate is contained to be phosphorylated, histidine 48. ATP is another substrate that can be at this domain. P2 helps bring other substrates to be in range to be phosphorylated, such as Ky or Kb. P3 is where dimerization occurs. P4 contains the active site for the substrates. And lastly, P5 is a regulatory domain and allows incorporation into signaling complexes. Figure 2 shown here is a substrate concentration versus velocity graph. And it was created to show the difference that binding to a signaling complex has on Ka, kinase phosphorylation, and the effect that the presence of an attractant ligand has on phosphorylation. Based off of the data received, the researchers found that the kinase in the presence of signaling complexes greatly improved phosphorylation, while the presence of an attractant ligand greatly reduced phosphorylation. Tables 1 and 2 related to figures 3 and 4. Figures 3 and 4 are simply bar graphs of the data presented in tables 1 and 2. Tables 1 and 2 are simply the kinetic parameters of P1 phosphorylation. The apparent catalytic rate constant, Michaelis constant, and the calculated catalytic efficiency are all shown for the phosphorylation reaction. The Kcat values were calculated from the Michaelis-Menten equation for saturation of both substrates P1 and ATP for each Kn determined by the experiment. Figure 3 illustrates that Kcat is increased 300-fold and is a primary parameter affected in kinase activation. Km was decreased a moderate amount, only about 3.3-fold for P1, but not much difference was observed at all for Km with ATP as the substrate. Similarly, in figure 4, it illustrates the comparison of kinetic parameters and the presence and absence of tar ligand aspartate. The value of Kcat was reduced by 40-fold with aspartate at 88% receptor saturation, and Km increased 2.1-fold and had no significant change for Km when ATP was the substrate.
Figure 5 displays the activation of the Ka P3-4-5 domain in core complexes compared to that as in small arrangements or small arrays. Both preparations are similar in activation from incorporation into a signaling complex and inhibition from ligand occupancy. So the essential conclusion from figure 5 is that we don't need to consider a local accumulation of substrate as a confounding variable that could also have altered the Kcat or the turnover number. Table 3 displays the data from the adaptational modification in small arrays or small arrangement. So note that there's only a threefold maximum difference in the Michaelis constant, but a large difference in the turnover number when glutamate is replaced with glutamine entirely. Figure 6 illustrates some of these comparisons pointed out in Table 3. The kinetics of adaptational modification are what's illustrated. So essentially, the kcats, the turnover number, and the kms, the Michaelis constants of the signaling complex, has a density of glutamates versus in adaptational modification when the glutamates are replaced with glutamins. Complete modification from glutamate to glutamine revealed an increased turnover number and a slight decreased Michaelis constant. And this increases efficiency because efficiency is defined as turnover number divided by Michaelis constant. Figure 7 illustrates the kinase activities of an isolated key A P1 domain versus that of an intact key A with all of its domains, as illustrated in Figure 1. The kinase activities were relatively similar, and the kinase activities of isolated key A P1 were variable depending on the key A's presence or absence within the signaling core complexes assembled with TAR, as well as whether or not aspartate, the attractant ligand for TAR, was present. So changes in the turnover number of the phosphate transfer to histidate 48 on P1, more so than changes in ligand occupancy or adaptational modification, has been implicated to be responsible for both the activation and the inhibition of the autophosphorylation reaction of P1 of key A. However, the implicated change in the turnover number refers to the P1 autophosphorylation reaction, ATP plus P1 to ADP plus P plus P1, and then phosphorylated P1 with ADP, but not the latter transfer of the phosphate to response regulators such as key Y. It may be possible that there is some impact of kinase control at the second step. However, this would be minuscule if even there. According to Table 1, the catalytic efficiency for substrate P1 increased by a factor of 1,000, and for the substrate ATP increased by a factor of 350. However, the researchers estimate that the kinase activity of the next reaction involved in cell migration, the phosphoryl transfer to key Y instead of key A, would not increase by more than a factor of 2. However, the sequestration or the accumulation and the gathering of P1 at a distant location, unrelated to the active site, does not contribute significantly to the control of the kinase. Images of sagittal sections of the signaling complexes have been interpreted to suggest that when the kinase is inactive or has very low activity, P1 and P2 are tethered on the key A, or sequestered. However, this study observed differences in activity by factors of 2 or 3 of liberated, non-sequestered, isolated P1 when reacting with the catalytic P3, P4, P5 region, suggesting that there is another major contributor to kinase control. Magnitude of activation is less impacted by ligand occupancy and adaptational modification, though this factor is still there to be considered. This is because physical contact between the kinase and the signaling complex can slightly affect the turnover number, or the kcat. Ligand occupancy inhibits the kinase phosphorylation. Covalent or adaptational modifications activate the reaction. For example, if negative glutamates are replaced with neutral glutamins at the receptor methyl accepting site. If the TAR contained pure glutamate as opposed to glutamine, activation was reduced. The kcat, or the turnover number, was only enhanced 3-fold as compared to 470-fold. And the catalytic efficiency was only enhanced 10-fold rather than 1,000-fold, as illustrated in Table 3. The Michaelis constant values, the concentration of the substrate, P1 or ATP, when the velocity is at half maximum magnitude, are within a normal physiological range. The relative consistencies in the turnover, or in the Michaelis constant, the Km, only a maximum of 3-fold difference, suggests that the dissociation constant is not the primary factor in determining the autophosphorylation of P1. Now, while the majority of the data is supported by previous studies, one kinetic analysis done of isolated key A in the P3, 4, 5 domains measured a 10 times higher turnover number for the intact enzyme and a 48 times higher turnover number for P3 and 4 and 5, and the Michaelis constant for P1 as its own substrate were 20 times lower than this study's analysis. However, there were a few differences between these studies that could explain the discrepancies. To begin, that study did not use intact signaling complexes, but rather key W complexes or coupling proteins. Also, that study's key A lacked the periplasmic or the transmembrane domains of the proteins. And further, that study's chemotaxis proteins were derived from a completely different species, from Salmonella enterica instead of E. coli. So the three-dimensional structures of these proteins were different.